Our next speaker is David Wild. He's an assistant professor at Indiana University School of Informatics and Computing. And he is a leader in the field of chemical informatics. And his research is across that chemical informatics and bioinformatics.
Great. Thanks, Judy. Well, thanks to Judy and Jeffrey for organising this, and it's great to have a chance to talk. So I'm going to be talking about big data in drug discovery. Now, drug discovery or the pharmaceutical industry has been in the news quite a lot recently for a variety of reasons. You'll see all kinds of articles about the healthcare plan, and we're not discovering new drugs, and where are all the new drugs, why are drugs so expensive, prescription drug pricing, that kind of thing. And it's kind of a history and a reason that drug companies have been in the news recently. And underlying a lot of those stories is the fact that we just seem to have hit a bit of a brick wall in terms of healthcare and finding new drugs and finding new treatments for diseases, for important diseases. So this is the area in which I'm working, and I'm really excited about the impact that informatics and computing, and particularly very large-scale data mining, can have. I think of the drug industry, or just the whole area of discovering drugs and medicines, as having some very kind of definable epochs. So arguably you can argue about where the field of drug discovery started, but the idea of pharmacy and having medicines and chemicals that you ingest into your body to make things better goes back extremely early, back into antiquity. But in a recognizable form by today's standard, it probably goes back to about 754 when the first pharmacy opened in Baghdad. And actually Iraq was a big instigator of the idea of medicines and pharmacies and having these bottles of things that you take to make you better. But the interesting thing is that process of drug discovery, we discover these things, we discover these medicines, these liquids or these solids or these plants that can help make you better. That kind of way of thinking about drugs really persisted up to about the 1950s and 1960s. And that's what I call the empirical epoch in drug discovery. It's all about just serendipity, about discovering new medicines almost by chance. Maybe you'll find some tribe, or go to China and find some people have learned to take this herb or this leaf or this plant to make things better, and that's your basis for treating the disease. Empirical evidence that it has some impact on it. Now, of course, it kind of advanced through time, and there was actually a real boom in the late 1800s where what we think of now as the pharmaceutical industry really kind of developed, and most of the major pharmaceutical companies now had their origins in the late 1800s. But there was a big explosion in the 1900s, kind of the mid part of the century, in finding new treatments. All kinds of important drugs which we now take for granted suddenly were discovered then. It's kind of partly because of the large investment that went into drug discovery, and partly through some of the processes which were able to extract the important chemicals in those plants or whatever they were, and refine them and actually make them on a large scale so that lots of people could take them. But we got insulin, penicillin, the pill in the 60s. Between about 1900 and 1960, we saw all these really, really important new drugs coming along. Antibiotics have made such an incredible difference to the world. So that kind of led to a very kind of strong pharmaceutical industry and strong tradition of research in academia related to diseases and medicines and such like. Now, there's this shift in the 1960s, probably the late 1960s, that peaked in the 1980s, which is what I think of as a second epoch, which is rational drug design or rational drug discovery. And this was when we started to get some real deep molecular understanding of disease and the impacts of chemicals, i.e. drugs, on those diseases. So we got in the 50s, we understood the structure of DNA, then we started to understand the structure of proteins and how proteins related in pathways, and we could start to think, okay, we have this protein here, we know something about this protein. We know this protein's involved in this disease. We're going to try and find a chemical that's going to stop this protein acting so that it can, we can help cure this disease. Probably the biggest success of this whole approach was HIV. So a vast amount of money was invested in HIV research in the 1990s. And it was the really first time there was such a massive effort put into the rational drug discovery. And this process involved understanding the proteins that were involved in the HIV virus. And there were three really important ones initially: reverse transcriptase, protease, and integrase. We were able to get crystal structures of those proteins, so we knew exactly the arrangements of atoms and bonds in those proteins. And then we actually developed chemical compounds, made brand new chemical compounds which were able to bind to those proteins and stop them working. And it kind of worked. Now we have protease inhibitors, reverse transcriptase inhibitors, which can keep AIDS at bay. We haven't cured AIDS, but we've made a major impact on its effects. So this was a big success. At the same time while this was happening, we had some really big new drugs come out. We started to get drugs for diabetes. Cardiovascular drugs was huge. Lipitor was the peak of this, was $8 to $12 billion a year for Pfizer. Everybody here must at least know somebody who's on Lipitor. Okay, so we've got these huge blockbuster drugs coming out, and most of these didn't actually necessarily come quite from this rational approach, but this idea of understanding all the molecular basis and these proteins and trying this lock and key approach of finding compounds which hit the target, and was certainly associated with that success. This kind of led to what I think it was the third epoch, which is the big experiment epoch, where we started getting a big shift in machinery, in actual technology, which could make biological experiments which used to be on small numbers of chemical compounds or small numbers of proteins and scale it up massively. So some examples of this were combinatorial chemistry. Rather than just synthesizing a few new chemicals at once, we could make hundreds of thousands of new chemicals with a building block approach of putting things together. High-throughput screening was a big one. And this was actually when I worked in various capacities for about 6 or 7 years in the pharmaceutical industry, right through this big experiment time. So one of my jobs was, I worked at Pfizer, and they just spent, I think it was tens of millions of dollars on this new machine called a high-throughput screening machine. And instead of testing, maybe 5 or 10 or 50 compounds at once for a particular biological action, action against a protein target maybe, it could test 100,000, later 1 million compounds. So literally they had this huge room with tiny samples of all the chemical compounds Pfizer knew about, which at that time were about 500,000 compounds. And robots would take all these and plate them onto wells and put them through machines and test them. And this was going to amplify this breakthrough. Lipitor was going to be a small fry compared to what we'd be able to do with this, because we could test all our compounds against all the diseases. And we can make hundreds of thousands of new compounds and test those and circulate this around. So it all became about numbers, about huge experiments. On the biology side, we've got microarray assays where we could take whole genomes and test compounds for their impact on whole genomes in tissue samples. Then we've got the Human Genome Project where we're going to actually understand this massive success. We now have this sequence of the whole, at least the significant parts of the human genome. But it still hasn't resulted in new drugs. If you look at the number of new drugs which come to market over the last 20, 30 years, it's drooping right down. The investment is going right up. The R&D spending of pharma is going right up. The number of technologies, the number of compounds we test, the number of assays, the number of biological results go up, but the number of new drugs is going right down. Now, there's all kinds of reasons for this, which I can't go into, but one of them is that what I observed when I was in the industry there is that this big experiment approach was all about generating data. So we have this high throughput machine. We can get hundreds of thousands of new data points in a day from one of these machines. Microarray assays, we have 14,000 genes, and we get data points for hundreds of thousands of compounds. We then had data on which kind of crossed different areas and such like. So, yeah, a big worry at the time is how do we store this stuff? How do we access it? Do we put it into Oracle databases? How do you find this? And the big struggle was with, how do we handle this data? But not what do we do with it? And still all this data has tended to sit in completely siloed databases. So we'll have a database of chemical structures, a database of genes, a database of bioassay results for this particular area. So we can kind of nominally access it. But we can't really do a big complete biology-wide analysis of it. Now, so this leads to what I consider, in my opinion, is the next phase of drug discovery, and that's the big data drug discovery, which is centered on informatics. Why do I think this? Well, firstly, we're just getting incredible amounts of data now going into public domain. Data that used to be in drug companies is now splurged out there in the public domain. We'll be talking about that in this talk. Secondly, this rational approach, which we've been kind of relying on, this idea of locks and keys. We have a protein target, and we're going to find a compound to inhibit this protein target. Very occasionally it works. It worked for HIV, and it kind of worked for a few other kind of what we call low-hanging fruit, the easy problems to solve. But if you listen to people in the pharmaceutical industry and in the biomedical communities in academia now, there's a little bit of humility coming into this whole thing and saying, oops, the human body is rather more complex than we thought previously. All these big breakthroughs like the Human Genome Project haven't, they've been had an incredible impact, but part of that impact is showing us how incredibly complex life is. It's a little bit like in physics we used to have the Newtonian model of the atom, or the Bohr model of the atom, all nice and simple, electrons going around. And then Einsteinian relativity and quantum mechanics came along and it's like, oops, this thing's a whole lot more complicated than we thought. And now the same thing's kind of happening in biology. So, for instance, we've had, people, vice president level people in pharmaceutical just admitting this. We don't understand biology. So we also don't understand how to turn off our cell phones before a talk either. We don't understand biology. We just don't understand the mechanism of disease. We don't know what's going on with most diseases. It's all incredibly complicated. So I think the important things here is that drug discovery is going to be informatics driven. It's going to be taking all the wealth of this data from different places and trying to make sense of it. It's going to be accepting the body is amazingly complex and we don't understand it well, and that everything's connected to everything else, which we'll get to in a bit. And this is just an example. I'm not going to take 10 minutes over each slide, by the way. So this is, just one example. These are known metabolic pathways of a single cell. And each of these, this is just blown up a bit here. Here's particular chemical pathways that are going on. It's amazing what we've done. It's amazing we can produce these maps. Huge success for biology, but we're just at the tip of the iceberg with this whole thing. Now I'm going to see if I can show you a little video now. This is slightly self-indulgent on my part. This was an animation done at MIT with a narration. And this is just a short YouTube video about the process that goes on in one of your cells when a foreign body comes along. So we've had a little signal sent out saying, oops, there's something here that shouldn't be here. We have to go and kill this cell because something's going on. And this is just a really nicely done animation. And the purpose of this is just to remind us of how complicated and amazing this whole thing is. Kinesin is my favorite, by the way, for this. Okay, let's go to, no, I think I'll get this right. We'll go to meeting controls. I'll stop sharing my desktop. And then we go to share web content. Now you should each see this on your separate computers. Let's see if this works. Okay, I'm just going to pause this for a second. We've got some audio with this. The audio is less important than the video for this one. Oops.
That finds an infection and goes through the wall of an artery in order to stop that infection. What you see now is called a lipid raft. It's made up of special phospholipids that pack tightly together and hold together the membrane proteins that are specifically linked to that lipid raft. These lipid rafts move around in the phospholipid bilayer and coordinate activity. Now we are zooming down one of the microvilli inside of one of those microvilli protrusions. What we see now is the polymerization of actin from the actin monomers, and actin forms the cytoskeletal network. This is gelsolin. It cuts off a part of the actin filament. Now we see microtubulin assembling from tubulin monomers. And this is called catastrophe, when the microtubules move back. This is kinesin walking along the microtubule and pulling behind it a vesicle. And along this picture, you can see a lot of microtubules, all with vesicles being pulled by various motor proteins. And in the center is the centrosome with its centrioles. This is the cell surface of the nucleus surface with the nuclear pores, and mRNA is coming out of the nuclear pores and now is assembling with a lysosome that allows the mRNA to be processed before the ribosome, which is shown there, begins to read the mRNA and produce a protein. This is another ribosome assembling onto the surface of the endoplasmic reticulum, or the Golgi body, and in this case, it's producing a protein that will be secreted. It's contained inside of a vesicle, inside of that membrane coat, and pulled to the outside of the cell by such a motor such as kinesin, which is shown here. These are vesicles that are being dragged from the Golgi body, which is shown right here, to the outside of the cell, and you're going to watch them being secreted into the space between the membrane and the extracellular matrix, which is above. These proteins are secreted, but they don't leave the membrane because they're bound to the membrane. And these are the proteins, the ICAM, that can reach up and bind tightly to the receptor molecules on a cell that is signaling a problem somewhere behind the cell. Once ICAM binds, it allows the white blood cell to go through a conformational change where the actin matrix dissolves, and it allows it to slither into the region where it needs to eat up the bacteria or the infection. And that's all.
So just share my desktop. Yep. Oops.
This is the story of a white blood cell that finds an infection and goes through the wall of an artery in order to stop that infection. What you see now is called
a repeat of it going on.
It's made up of special phospholipids that pack tightly together and hold together the membrane proteins that are specifically linked to that lipid raft.
Yeah.
These lipid rafts move around
Yeah.
in the phospholipid bilayer and coordinate activity. Now we.
Okay, good. Oh, we have another one going.
This
Good.
is the story of life.
What do we do? Just desktop. Okay, good. Video was so good we wanted you to see it 20 times. I also think that's a nice video to watch if you ever don't like the way your hair looks, or you're not so happy with how your body's working nowadays. What's going on at the cellular level, at least, is quite awe-inspiring. Okay, so let's get into a bit more detail about what this data actually is. Well, it's only recently that all this data has been there. But there's now what I think of as an incredibly rich resource of information in the public domain. It's all free, it's in databases you can access, download, connect to yourself, relating to all these things that are to do with diseases, chemical compounds, protein targets, genes, pathways, diseases. So here's some examples, and these numbers are kind of got together last couple of months, but they're already out of date. 69 million compounds. You have information in a database called PubChem on 69 million chemical compounds and 449,392 biological assays. A biological assay is something where a bunch of compounds are tested, say with a high-throughput screening machine, for activity of a compound against a protein target or a cell or a gene. So it's something which links a chemical compound with a cell, a gene, or a protein target. There are 4,763 known marketed drugs, 9 million protein sequences in SwissProt, and 58,000 three-dimensional structures of proteins in the PDB, 14 million human nucleotide sequences, 19 million life science publications. A whole lot of the knowledge is embedded in publications. When I checked, about 19 million life science publications in PubMed and about 800,000 every year going in there. That might not seem a big number compared to the rest, but think of the complexity of a paper. You can't read 800,000 papers a year. But even more important than these entities themselves are the relationships between these entities. We have a whole wealth of ways that these entities could be related to each other. Biological assays, as I said, gives a number which is an activity of a compound against a protein target or a gene or a cellular assay. A crystal structure of a ligand-protein complex shows how a chemical compound binds to a protein target. Two things are mentioned together in a paper abstract. Well, that's some kind of relationship. There's some reason two people have, these two things have been mentioned, a gene and a compound, are two genes in a scientific publication. Computational experiments. We have all these predictive models and docking models and all kinds of things that can infer relationships between things at a computational level, and they're another way of making a connection. Statistical relationships, we find them referenced in different ways. System association, you remember that, what I just showed. There's a systematic, a plumbing relationship that we know about between these two things. So just an example with this PubChem database, how recent all this is. Back in 2005, there were only 2 million compounds, 2.8 million compounds in PubChem. We're here now at 2010, we have 69 million compounds. Number of bioassays, this thing jumps up in leaps. Until a couple of months ago, we only had about a few thousand actually. This is about 6,000 something, 60,000, jumped up to 484,000 bioassays. So these things can suddenly, somebody puts in a new data set and they jump up. Now the 69 million compounds, this is just an example of a PubChem page for one compound. This is the one for ibuprofen. It goes on and on for several more pages. All kinds of information about synonyms of this compound, what it's known to have been tested and active in, relationship to protein targets, chemical properties of the compound, and so on. Okay, sure. Chemical properties and so on. So each of these 169 million entities has all kinds of connections to other things. Look at proteins and genes have gone up even more impressively. This is a log scale on the left. So we've gone from 10 to the it's kind of hard to see. Let's look at just the number of SwissProt entries, for instance. We've gone from 10 to the 3 up to 10 to the 8 expected for 2010. So, what I'm really interested in is mapping, being able to look at all these connections between all these things and then extract real science out of it, or something which real scientists can use for knowledge discovery. We'll talk about this in a little bit, but this is something we've done called Chem to Bi to RDF, which maps a lot of these links between chemical compounds, drugs, protein targets, genes, and so on. And this is actually just a list of the databases there, the number of entities in the database, the number of relationships between each of those data sets. This is not something which has been mapped until recently. Each of these databases is siloed. So you go to a different website, you can search this database, but you can't in a single resource until now be able to see all those relationships together. Now we haven't finished talking about the amount of data here because you're a big pile of data as well. There's something called next generation sequencing coming along, which you may have heard about, which is the ability to sequence your whole personal genome, not the human genome in general, but all the little quirks and ups and downs and changes and what we call SNPs, different alternatives for gene positions that you have in your particular body. And that comes to something like 6 billion base pairs, I think, or something. A vast amount of data in your body, in your genome. But you've also got a vast amount of data in your body as well, how your body reacts to certain things, what you're allergic to, what drugs you're taking, what conditions you've had previously, where you live, what toxins you're exposed to, how old you are. All these kind of things play into how your body reacts to certain things. Now, we're just starting on this, but this is kind of the genesis of a whole area of when all this next generation sequencing comes on in the next few years, when we're gathering all these data in patient health records and electronic health records like Google Health, or this one, Patients Like Me, how do we connect all this data to the molecular level? Can we use this to understand you at the molecular level based on what makes you unique and what we understand about all these connections between things? There was an interesting bit of work that was actually published in Nature. Unfortunately, my publications haven't quite got as far as Nature, but one of my colleagues did this really nice piece of work where they're looking at known drugs and protein targets. And you actually started to infer relationships between drugs and protein targets just by looking at how similar they were to other drugs and the drugs which protein targets, those drugs related to, and such like. And they made these big networks of inferred relationships. So this is, just based on looking at the similarity of the structures and similarity of the targets, can we just fill in get some of the lines? We already know some of these relationships. Can we infer some other relationships based on that data? The nice thing about their work, which is why they made it to Nature, was they actually did the experiments. They went away and took a bunch of these inferred relationships and tested it, and they came out with a whole bunch of new real experimental data based on that. And there was, I think, 3 or 4 of them where there's a really significant advance in knowledge just based on doing that analysis and then doing the experiment. So this has led to a kind of a new field which is tentatively called systems chemical biology or chemogenomics. Chemogenomics is a little more restricted, looking at the relationship between chemistry or chemical structure and genes. Systems chemical biology is saying, let's look at the whole picture of what happens when you put a drug into the body. It's a hugely massive problem, but can we look at that whole picture? And there's a really nice Nature Chemical Biology paper on this by Tudor, Opria and others a couple of years ago, really introducing this field. So this is the area that I'm working in and really excites me based on what I know about drug discovery and what I know about what's going on in data mining. And what I think is exciting is the kind of confluence is a whole set of technologies coming together that mean we still have an incredibly big problem in trying to understand the human body from an informatics and data perspective and being able to turn that into knowledge that scientists can use. But we've got these technologies coming together that where we can scale up what we're doing to a level that we can start to tackle some of those problems. One of them, which we all know about here, is cloud computing. We can do data mining on a vast scale now. We can scale up data mining. So we have these predictions and methods and querying where we can, we used to be able to do it on a small number of compounds. We can now not just do it on compounds, we can do it on relationships and connections between things and expand right out and do it in real time. Now we're just starting to see, this is why Future Grid is exciting, we're just starting to see what's possible with that kind of technology. We then have what I call integrative cheminformatics and bioinformatics. We're starting to get the people together. We're starting to get the people together which relate, the cheminformatics people which know all about drugs and chemical compounds with the bioinformatics people who know all about genes and proteins and diseases. And we're able to find, use the other technologies together to make an impact there. We're starting to get this new field, as I said, of health informatics, where we're not just looking at the molecular level, but we're looking at the molecular level about you, and we're starting to get patient data coming in. And it's a field called quantitative pharmacology, which is related to that. And then we've got some, what I think are really important, kind of generalized semantic technologies, semantic web, and also complex systems, network methods. and tools, which we can do, enable us to do what I think of as human scale data mining. So, human beings can only look at a certain number of things at once. They can't look at 69 million things at once. But we can do things like projecting down numbers of dimensions. We can have networks where we emphasize certain parts of networks so we can visualize them. So we can go from this huge network of everything connected to everything else to something which is manageable for a human to interpret. Then this on the right is kind of these layers that I see it as. So, before we can do anything else, we have to be able to access all the data. And that's been a big problem in the past. We've mostly solved that with the exception of next generation sequencing. We still don't know how to store and access next generation sequencing data effectively because there's so much of it. Integration we've kind of solved now thanks to RDF and semantic methods in general, and also graph methods as well. Now, there's still a lot of problems with it. There's problems with ontologies and making ontologies work and making all this work for us, but we've kind of pretty much solved that problem of bringing it all together. And that's what we've done in some of the work we've done at Indiana. But the next stage is analysis. It's taking all these things of everything connected to everything else and making it into something useful for the scientists to discover new drugs. And that's a kind of analysis process. And we're looking at ways, tools for doing that, ranging from highly visual tools that kind of enable you to see the whole kind of Google Earth kind of view of what's going on, down to kind of querying and hypothesis generation tools that allow you to actually try out different hypotheses. So, we're not the only ones working in this field, but being a professor, I get to blather on about my own research more than anything else. So this is what we're doing in this area. It started off a few years ago when we developed something called ChemBioGrid, which was about the access portion. So this was the whole set of chemistry and chemical databases and computational methods for chemistry, like molecular docking and predictive methods, which weren't integrated well at all. You couldn't access these things in a kind of a seamless fashion. So we developed this thing called ChemBioGrid, which was really a web service infrastructure for cheminformatics. So this is bringing everything together in one framework. So we can take a computational method here and apply it to some data from over here, or we can take some data from this data set and bring them together. And this was really interesting because it spurred a whole bunch of what, back then mashups were the big thing. So we had a whole bunch of mashup tools we were able to develop, which one nice example was user scripts in Firefox web browser. We were able to develop scripts that when you went to a publication page, it would automatically highlight the chemical structures in there. They could shell out from those chemical structures to information from PubChem and searches and all kinds of things. So that was step one, which is kind of access. And we then got into the semantic web the purpose being to look at integration, but not just integration, i.e. connecting everything with everything else, but doing so in a meaningful fashion, i.e. a semantic fashion. So here we start to look at technologies like RDF. RDF is all about mapping those relationships. OWL, which is all about ontologies, so it's specifying the meaning of those relationships, and then Sparkle, which is for querying the relationships. Now, I feel like these technologies weren't really useful until a couple of years ago. For some reason, it didn't quite all work together. But just in the last couple of years, we've really been able to make these technologies work for us. So this on the right here is RDF. This is just saying an RDF triple, it's called. It's really just a noun, verb, noun piece of information. So it's this entity is connected to this other entity, and this is the property that connects them. So this chemical compound is active against this protein target, or this gene is part of this biological pathway. And a lot of those are quite simple relationships. Some of them are very subtle and complex, so active against can have all kinds of meaning, but just mapping that association becomes very useful. So we made this resource called Chem2Bi to RDF. We just published a paper in BMC Bioinformatics on it, which really realizes that RDF level integration for chemical and biological data together. Now, in its own right, just connecting everything with everything else isn't necessarily useful. But one immediate thing we're able to do with it is to, and this is just the context of where it is, so we have a triple store in the middle with all this data in, and then we connect out to all kinds of other data sets, and we have all kinds of tools which use that, which we'll look at. Okay, I think we're going to skip over some of this. So we went through a fairly standard procedure for RDFizing, or converting this data from databases into RDF. And we've also linked into things like the linked open data cloud, which is a big effort to link everything together. So we've pushed our stuff into, you can go to linkeddata.org. Our stuff is now all in linked data. So we're not a resource that's walled off from everything else. So we have the power to decide what's in Chem2Py to RDF, and then that becomes a silo from everything else. Once it's in RDF format, you can just throw it into these other resources in the linked open data cloud, and it can become linked with everything else. Now, there's a lot of subtleties, and it's not that easy in some ways, but in some ways it's amazing how straightforward it is to do that. So, one thing which is, sounds simple, which we can do with Chem2Py to RDF, is simply querying. Just like you use SQL to query a Postgres or an Oracle database, we can use Sparkle to query these RDF relationships. But the really interesting thing here is those relationships. So we're not just querying one data set, we're querying this whole network. And this enables us to do some kinds of scientific querying, which before were almost impossible to do. They was possible, but nobody really did them because they was too complicated to bring the different kinds of data together. Here's an example. So these biological pathways are involved in all kinds of biological processes and disease processes. So often we want to inhibit particular genes or the protein products of genes in a biological pathway to disrupt that pathway. Or maybe the pathway is being disrupted by disease and we want to get it going again. But the problem is a lot of these pathways are redundant. So if you think of a pathways, literally pathways, all these stages through it, like those metabolic pathways we saw, so we hit this protein target, the lock and key model. We're going to take out this protein to, there's this, hyperventilating pathway that shouldn't be doing it, but because of this disease it is. So we're going to hit this protein and stop it working. Unfortunately, this robust, it just takes another path round by something else. So there's a lot of interest now in multi-target inhibitors, chemical compounds that can hit multiple points in that biological pathway. Now this becomes a kind of difficult informatics problem because we then have to say, well, what compounds are there that can hit multiple points in here? Well, then we have to understand the relationship between all the compounds and all the different protein targets or genes within that. But we can do it now with one Sparkle query. So with one query, we can say, find me all the compounds that inhibit at least two of the targets in the MAPK signaling pathway. It's a very straightforward Sparkle query, and it comes out with a list of compounds out of the 69 million in PubChem that we know are capable of doing that. And we actually, mapped this back to some of the science with publications, and it bore out. Another area is polypharmacology. We know, this lock and key model, we're looking for this compound that inhibits this protein target, but unfortunately, or maybe fortunately, it does all kinds of other things as well. It hits lots of other protein targets. So we find a lot of drugs side effects are because of what we call off-target interactions. The drug's not only just hitting what we want it to hit, but it's interrupting various other things as well. So, say we've got a compound or a drug that has a certain profile across protein targets that we like, but certain other ones we don't like. We then want to do searches for other compounds that might share that profile, not just that one lock and key effect, but a whole profile across a genome or across a set of protein targets. Again, we can do this now with one query in Chem2Py to RDF by simply saying, find me all the compounds which have the same profile, or at least so many interactions in common, at least so many relationships in common, with, in this case, dexamethasone, which is a cancer drug. So even just simple querying, we can do things which we haven't been able to do before. And we actually now have some partners in the pharmaceutical industry that are using this to actually do some real drug discovery. But we want to go further. We want to see, what can we do to help us get a broader understanding of what compounds are doing and the relationship between genes and compounds. So I actually partnered with Judy Chu and Jeffrey Fox to use some methods they developed for clustering. So these are basically projection methods. So they can take multi-dimensional data and project them down into a smaller number of dimensions. So what we did was we took the compounds in PubChem, this huge data with 69 million compounds, and said, okay, we can calculate similarity between these compounds. We can plot them in a multi-dimensional chemical space, but that chemical space we had 166 dimensions. But we use the GTM and MDS projection to project it down to 3 dimensions. Then we have a kind of Google Earth for chemistry, so we can see the whole kind of span of chemistry in one view. And then we can launch these queries, like these queries that do, look for compounds that have certain profiles and certain relationships, and we can project them onto this view. So this is, we actually had a conference publication on this a little while back. Here we actually see, you can't really see the detail here, but this grand view we see here is of the, this is like the whole of chemical space. We have these interesting clusters of areas in chemical space, but the colors here represent certain relationships between compounds and genes. So you can start to see the relationships of how different areas of chemical space relate to certain associations with genes. One specific way we're using this is what I call like a Doppler radar plot. You know those maps of, the weather radar maps where you can identify a tornado because there's winds going away from the Doppler radar and towards it, so you get suddenly contrasting colours on the Doppler radar, and you can automatically detect a tornado. Well, here we're doing the same kind of thing, but this case it's kinase specificity. Kinase is a family of proteins involved in all kinds of processes in the body and also implicated in cancers. And there's a lot of compounds that interact with these kinases. Some of those compounds interact with only a few, and some interact with all of them or a lot of them. So we're interested in regions of this chemical space, or instances where we can have small changes in the chemical structure of a compound, which can make big changes in that specificity. So can we take a compound which interacts with all the kinases, which are generally a bad thing, make a few tweaks to it, and then it can just interact with 1 or 2 kinases we're interested in. So here what we're doing is we're projecting, this is a bunch of compounds, this is a kind of world of compounds, Google Earth of compounds that have some kind of kinase relationship. The blues on here are ones which only interact with a few kinases, and then the oranges and reds are ones which have this kind of promiscuous effect. And it's very hard to see on this display, but we can actually identify areas here where we get this Doppler radar effect, where very small changes in the chemical structure, a small distance on here is a small change in the chemical structure, make a big impact on that specificity. We go from a blue to a red, so we go from impacting 1 or 2 to impacting lots. We're also looking at network, what we can do with that network and that graph of everything related to everything else. And one of the key things is finding paths, and this is all about hypothesis testing. So all the time in drug discovery, people have hypotheses. I think these kinds of compounds might be useful for this disease, or might interact with this target. Or I think there might be a relationship between this target and this target, or this gene and this gene. What these methods we're developing now enable you to do is to just ask that question of this whole network. What are all the relationships between this gene and this gene, or this chemical compound and this disease or this target? We developed something called Chem2Bi2RDF dashboard, which literally lets you specify those 2 endpoints and look at all the paths between them. We've also got a nice tool that Yuyin here developed called Pathfinder, which actually does this in real time. You can actually, we could shell out and go to this URL at the bottom here, but you can actually see this network of paths, the relationships between things develop in real time. So on the left here we have actually here 2 related steroids, triamcinolone and dexamethasone, and this finds all the gene relationships. So this is just verifying. This just tells us what the scientists already know, but it's brought out automatically. The main relationship between these 2 steroids is they both interact with these two genes, the NF-kB1 and the glucocorticoid receptor. And this is well-known science. We haven't learned new science here, but we've verified the method with some well-known science. What we're actually doing now is we're looking at recent papers published in the Journal of Medicinal Chemistry, which is the main drug discovery journal. Very new research where people have found the link between two things experimentally, but that's not in the data sets yet. And we're going back to see if we can, if we can put that hypothesis in and find it through different ways. And we're also plugging into something called Cytoscape, which is a network tool. And again, this brings in the cloud computing aspect, because we want to be able to do dynamic querying. So we want to be able to click on this object here, this gene and this compound, and in real time see all those relationships and be able to filter them down in a reasonable way. And we actually have a prototype plugin that does just that, but we haven't got the cloud computing backend working yet to make it really fast. And then these are just a bunch of, we have a whole bunch of these. This is an interesting one where we actually, there was this paper published a while back. Was it this one or was it this next one? Yeah, this one. Actually found a previously unknown relationship between two chemical compounds that were known to have an effect on tuberculosis, and they discovered this thing called INHA, which is a gene which these compounds have a relationship with, and they were able to explain why both of these compounds are effective against tuberculosis. But we were able to find it too without the help of that paper, simply by finding these multiple path interactions between things. I'm going to skip over this bit. This is just another tool which is used at Lilly for this kind of thing. But a big part of this whole thing, as I said at the start, is journal articles and the scholarly literature. So much information is embedded in that. Now, some of that is humanly extracted and put into databases, but particularly the latest stuff isn't. It's just there in the publication. So we've started to put an effort into what I call semantic text mining of journal articles, not just pulling out entities, but pulling out relationships between entities. And we actually published this paper a while back where we were able to use a mix of natural language processing and named entity recognition and some machine learning to make a reasonable good job of, at least in this particular domain, of finding relationships, extracting relationships which were described in papers between chemical structures and protein targets. We're now doing some really interesting collaboration with some people at Tsinghua University in China, where we're doing something called topic modeling. So what we're doing is we're taking the whole of PubMed, all those 9 million publications, but many of the most recent ones, and we're extracting all the named entities, the chemical compounds and the genes, the diseases, the targets, and we're putting those into our network. So we found this is the PubMed ID of the paper. It has this word hepatotoxicity in it. We identify that that's a side effect, and now we relate that to a CIDR, which is a side effect database. It has a compound in it, and the ribapine, so we find that in PubChem and connect it to PubChem. We find these gene names. Gene names are quite easy because they're easy to distinguish from other things. But we relate this to Uniprot feed. So we plug the paper into this big network as best we can. But then what we're doing is something called topic modeling, where we're actually using a method that looks at all these relationships between these entities and build these little clouds, these little topic clouds of things that are related to each other. And now we can use these topic clouds to effectively start to rank relationships. So we're using something called BioLDA, which actually looks at, within this context, if two things are related, what the significance of that relationship is against the background noise of all the generalized relationships. So is this a really significant relationship we found, or is it, is this one thing related to everything? It's just a kind of washed out thing, or is this a really significant? And I don't really have time to go into the details, but it's able to come up with probabilities and these entropy measures related to that. So we're relating that with our methods to find paths between things. So we can find these paths and then rank the paths and order them. So then we then think of these paths not just as things in their own right, but pieces of evidence linking two things, linking a compound and a gene. And we can rank those pieces of evidence and cluster them together. So one of the projects we have going with Lilly is looking at, taking that and saying, okay, there's a bunch of evidence, there's a bunch of paths relating this compound with breast cancer, but what is that evidence? So we cluster the evidence, so well, we have these biological assays which we think are important, we have these paper journal articles which we think are important, but we rank them up, we bubble up the ones which are most significant to the top, and then, then we can go out and look at the less significant things later. So we're thinking in terms of evidence ranking, enabling a scientist to view the piece of evidence which seemed to be most significant relating, say, chemical structures or drugs to particular diseases and targets. So in summary, I would argue that drug discovery is entering a new area which has to be informatics-based, because there's so much data out there which is important, and the human body is so complicated, we can't use traditional kind of naive processes for being able to do drug discovery. And we have this new approach which of systems chemical biology, which is kind of codifying that. And I think by doing that kind of analysis on all these relational data, we can start to learn new science about how the human body works, how disease works, and how we go about treating those diseases. The semantic web is kind of neutral in all that, but it's a useful framework at the moment. 10 years from now, we won't be talking about semantic web, we'll be talking something else. But right now, it's a useful framework for implementing that. So I don't like to overhype the semantic web. It's just a framework for implementing those kind of relationships. But it provides a physical kind of way of doing that. And then together with cloud computing, we can have the computing power to be able to do all the computational necessary to pull these things out. And we've made some early steps in this. Again, I don't want to overplay what we've done, but certainly our contacts in drug companies and also in academia in the biomedical field who we're collaborating with are starting to find this kind of way of approaching it useful. We haven't discovered a new blockbuster drug with it yet, but it's starting to have an impact on the way people think about doing drug discovery. But we've got a lot to do. We have to really get this ranking worked out. Right now we have far too many paths between important things. So we can easily, I showed some examples where it's kind of restricted, but we can have hundreds of paths between things. How do you go about working? Hundreds is better than millions, but how do you go about making that more humanly manageable? We need to aggregate things better. We're going to have to pull in the patient data, the next generation sequencing, connect that to the molecular data and make some sense of that. So we're kind of flailing, we're making some steps, but the problem's getting bigger and bigger as we're making these steps to tackle it as well. But I think this is a really exciting field. We're making a small contribution to it, and I think we all agree healthcare is one of the biggest problems that we have to tackle nowadays, and it's kind of exciting for me as a computer person that we can maybe have a major impact on that over the next few years. Okay, thank you.
So let's thank David for his excellent talk about drug discoveries, and we can take some questions.
Penn State has a question. You talked about cloud computing and how you used it to solve some of these problems. I was wondering if you could be more specific. What exactly do you mean in this context when you say cloud computing?
Sure. So these are various ways it can come in. I'll give a couple of examples. One is the Virtuoso triple store. So right now we can quite officially handle single Sparkle queries on the data set, and we can reasonably efficiently, within a minute or so, we can find relationships between 2 points. But what we really want to be able to do is to click on 2 things in Cytoscape and immediately see the relationships, and then start knocking things out. No, we're not interested in this, this, and this. So we want this to be a interactive process rather than a kind of wait a minute for it to happen process. So a scientist can have a process of going through the discovery. So right now on the back end of it, we just have a Virtuoso triple store on a single machine. What we want to do is, there are various methods out there of making Virtuoso run on the cloud. So we run queries, separate queries on the cloud, so we span out a whole bunch of parallel queries and then collect them back in. And it fits the model really well because we can then look at multiple hypotheses at the same time, and those become embarrassingly parallel jobs that can be spawned off and brought together. So there's no reason why we have to process all these different Sparkle queries sequentially. Another example is with a visualization with the clustering methods. Right now we kind of impute new points in. We could go off and actually generate new clusterings on the fly. Now there's some barriers to do that from the algorithmic point of view, but I see cloud computing as firstly enabling us to do, you almost add a new level to it. So we can then do thousands of Sparkle queries and mine the results of those Sparkle queries rather than just doing one Sparkle query, if that makes sense.
So, sorry to interrupt. So you mean you haven't utilized cloud computing yet? You're planning to?
Well, okay, it depends how we define it. I mean, we've certainly done parallel processing.
I think that's kind of my.
Yeah, right.
I think that's kind of my point. How do we define the way you're using cloud computing in the cloud? I mean, what is that exactly?
I mean, we don't routinely run our stuff on Amazon, for instance. So we're not routinely right now utilizing cloud computing, but we are building our stuff in a way that you could do that. Do you want to say anything?
So I think the flexibility of that, and actually because we collaborate on this
Right.
project,
Judy's the expert in this.
so I can make some comments complementary to his comments earlier. So I think that it's because cloud is elastic, so if that we can use this system in a way that on demand is very helpful. Otherwise, we can actually run this this kind of parallel jobs on the cluster.
Good. Yep.
Minnesota has a question.
Okay, go ahead.
Hello, this is Elizabeth Sandberg, and I am in the Department of Family Medicine and Community Health, and we're working on a project called EPCRN, which is the Electronic Primary Care Research Network. We work with EHRs and ontologies. We're pulling all these tools together for our federated query system. And I have a question on RDF. We are looking at RDF and using it the way you are using it with a triple store. You were saying that there were issues that you had with RDF, and I'd like to know how you solved some of those. In the talk previously, I know that Johan Bolin mentioned RDF, and he said that they abandoned that idea and went back to a relational database. So I just would like your comments on that.
Right, so I should say I kind of smoothed over a little what we're actually doing. So what we actually have for Chemtabyte RDF is a relational database in the background, and we also have a triple store. And you can access it through either of those methods right now. But they both look the same on the surface. So you run Sparkle queries ostensibly on RDF, but one way it gets translated into a database query or set of database queries. In the other way, it gets done on the raw RDF triple store. One of the problems with the RDF triple store approach is that RDF is very verbose. So you get these, and you're storing all the so relational database stores things, and then there's ways of highlighting those relationships between things. A triple store actually stores the relationships themselves. So there are various technical reasons, which I'm slightly vague on, but I can remember if I really have to, about there's certain things you can do with a triple store, there's certain kinds of query you can do with a triple store that you can't do with a relational database. But I think the bigger problem on top of that is that RDF is only as good as the data you have in it. So, and that gets to ontologies. So, what's a chemical compound? We define a chemical compound as a PubChem ID. So that's quite easy to encode. We can have a little URI, because we call it, which identifies that PubChem compound. But there are other compounds which are not in PubChem. You could make a new compound which is not in PubChem. So how do you identify that? So we can get, sometimes it's easy to have an ontology which specifies the things, this is a compound, this is a protein target, and this is an interaction. Find particularly those interactions are very difficult to code in an ontology. So when we get to these high-level things like trying to rank relationships and understand the meaning of relationships, we can fall foul of having too vague an ontology or too vague relationships. So say an interaction. A lot of our relationships are association. So there's an association between this compound and this gene. That association might be in a paper, or it might be an experimental association. Those two things will be very different. We can have a negative experimental association where something was found not to be active. So we're now just trying to get into how to encode those things, and then you get into domain ontologies and making those domain ontologies work together. So you have this kind of slippery slope that you only want to go too, if you go too far down it, you get bogged down with experts trying to decide what's what. But you have to go a little way down that to be able to make things work well. So there's this kind of sliding scale with the ontologies that you have to work with, and we're kind of trying to work that through at the moment. There is actually a project here as well looking at kind of patient health records and how that can be put into RDF triple stores and so on. So we're kind of, working with that. My preferred approach is just to do it and then fudge along as you go along. So, rather than trying to solve all the problems now, which gets bogged down, you solve the problems as you come across them. The nice thing about keeping the relational database is you can always go back and do things differently. So you can recreate your triple store from scratch if you want to do things differently. Did that help at all?
Somewhat.
So is there any more question? All right. Okay.
Okay, thank you.
Thanks, David.